that is created by the Golgi complex. You get some proteins processed in the rough ER, they get sent down to the Golgi, they get further processed, and then the Golgi spits out an organelle. And this one's actually called the lysosome. So our lysosome is involved with housekeeping. It doesn't actually physically detox anything. So if I asked you, if you ate some poison berries, what would be detoxifying those? That would be the smooth ER. But any kind of waste that would be created from that might be cleaned up by something like a lysosome. So detoxification is actually up in the smooth ER, but cleanup after that process is in the lysosome. The lysosome is our housekeeping. And to do that, the name says lyse. Lyse means to rupture or break apart. So our lysosome just has a whole bunch of nasty enzymes in there, and enzymes are proteins, that just like to break stuff apart. That's their job. They chew stuff up, they spit it out. There's about anywhere up to 40 different types of nasty little enzymes called lysozymes that can reside there in that lysosome. Depending on the cell type, what it's eating, what it's doing, what it needs to clean up, you'll use a different complement of enzymes. But all these enzymes work pretty much the same way in tearing stuff up. They also work in the same way that lysosome, inside of it, is very acidic. If I went out the cytosol and grabbed a pinch of that and checked the pH, 7.4, 7.6. If I go into the lysosome, pick out a little bit of that material in there, you're talking about pH 4, pH 5. For a cell, that's pretty acidic. That acidic environment helps to break things down, but it also has a second function as well. So this lysosome has the ability to pump in hydrogen ions, keep the environment nice and acid. If you think about what a cell's involved with every day, if I drop a book on my foot, those cells are going to get disrupted. It's possible that the contact of something smashing into my foot could damage organelles inside the individual cells in my foot. I would need to replace them, or they would need to heal. It's possible that some of those organelles that get damaged could rupture and leak out different contents. Well, what would happen if some of these nasty little lysozymes got loose in the cell? Their only job is to chew stuff up. Are they going to be too discriminate about that? No, they're just going to go and eat stuff. So that acidic environment, on top of helping to break stuff up inside the lysosome, is also a control mechanism, because the enzymes that are in there won't work at the regular pH. They only work at that acidic pH. If they get out into 7.4, they don't work anymore. It's kind of curtains for those enzymes, which is pretty interesting, because you think, well, neutral is neutral. Everything should work at neutral. But not these proteins. These proteins like acid. It's a control mechanism, so that if they get loose, they don't do unnecessary damage to the cell. So after these cells, after these lysosomes, after these organelles go in and do their job, there's broken down bits and pieces of stuff. Now, some of that can be reused. Just remember, what is everything going to be made up of? Well, those 4 biological molecules. And what are those 4 biological molecules? We have carbs, nucleic acids, proteins, and our lipids. All of those are organic, which means they have carbon skeletons and hydrogens on them, and then assortment of other stuff. They're all made out of carbon. Some of that stuff we can reuse. We can make other carbon skeletons from them. So if it's possible, the cell really is pretty efficient. It will try to recycle as much material as it can, because a lot of those materials are made of the basic building blocks. Anything that it can't, that's too worn down or too messed up or just not the right thing to be able to rebuild with, will get disposed of. It'll get shipped outside of the cell. It goes into your lymphatic system, and then that recycles, goes into your kidneys and into your colon, and exits the system through your waste. The other kind of secondary job that a lysosome has is to digest anything that comes in that shouldn't be there, especially something like bacteria. So these have a job also in immunity. You'll see immune cells, like you have macrophages and all these immune cells in your body. Those cells tend to have a lot of these structures. So remember we said different cell type or different types of cells have different proportions of different organelles. Something like the macrophage has tons of lysosomes and helps to break down bacteria and get rid of those invaders. And speaking of invaders, now we're going to talk about our last two major organelles. So everything we've talked about has been like housekeeping or how to prep a protein or kind of structural, general running of the cell. But all those things require energy, and that's where we're going. Our next chapter is about energy, and we're going to talk about how a plant gets energy and how you and I get energy. There are very specific organelles involved with those processes, and that is our chloroplast and our mitochondria. So we're going to start with our chloroplast. This is specific to plants and algae. It's typically where we find them. They also, for the plants and algae, are going to be the source of food for that organism. Those organisms are said to be autotrophs, which means they're self-feeding, which means this little structure will make their food for them. Now, it doesn't mean that structure can get the energy out of that food. It just means it makes the food. So even our plant is going to have our mitochondria, which is what we're going to talk about next. But this structure, this chloroplast, will make that food for the plant. So it doesn't have to go out and hunt anything. It doesn't have to go out and get food. Most of the food, the sugar foods, are made right there for the plant. Now, if you look at this structure, there's a little picture here and it shows a little kind of the TEM image of it, and then there's a little cartoon image of it, and they've got some stuff labeled in there. If you're looking at that picture, something should kind of jump out and grab you that doesn't look like it belongs there. So kind of think about something I said earlier in the lecture. And look at that picture for a minute. Kind of think about what jumps out as odd inside of this chloroplast. And hopefully what jumps out at you is that there's DNA in there. Well, DNA belongs in a nucleus. Why is there DNA in a chloroplast? Oh, what a great question. I'm glad I asked it. Okay, so we look at this DNA. You can see it's the little blue circles that are labeled inside of that. If you look at your textbook, the same pictures in there, you might get a little bit better image of that if you look in your textbook as well. That DNA doesn't look like the DNA in the nucleus. As eukaryotes, we have what's called a linear chromosome. And we, as humans, have 46 of those. Any one of those 46, I can grab it each end, I can pull it out straight to a line. This thing is a circle. That's a circular chromosome. That doesn't look like anything that's in our nucleus. That's because that's not what we find in our nucleus. That type of DNA, that circular DNA, is actually found in prokaryotes. Remember those things we said we're not going to talk about too much more for the rest of the semester? They actually have what's called a small circular chromosome. And the DNA in a chloroplast looks very much like a bacterial chromosome. Also, we have DNA in here. We also have a ribosome in here. Well, it kind of makes sense. We're going to have DNA. It means we're going to probably make RNA, and we're going to make some specific proteins. We're going to need a ribosome to read that. This ribosome's a little weird also, though, because this ribosome, for any of the organelles, kind of one of the things we can look at them is tell how they function is what size they are. So there's a really clever way to figure out how heavy something is. They throw it into a liquid and see how fast it sinks. If you throw a boulder into a river, how fast is it going to sink? Now, if you throw some sand into a river, how fast is it going to sink? Same thing with organelles. If I throw something big and heavy like a boulder into some liquid, it's going to sink really fast. If I throw a few grains of sand, it's going to sink slower. Ribosomes are very nonspecific. I could take any DNA, RNA from anything, stick it up to any ribosome, and it would read it. It wouldn't care. I could take my DNA, my human DNA, put it into a bacteria, and that bacteria would make my protein for me. It's actually how they make a lot of human insulin these days, is they have bacteria make it, because they're like little factories. We can put human DNA into the bacteria, and it'll make us tons of insulin that we can then use for diabetics. And it's clean, and It's a nice source of clean insulin. But just because they're non-specific doesn't mean they're exactly the same size. A bacterial ribosome is lighter. If I pick it up and I throw it into